talks today. If it's your first time here, we're going to have two 20-minute talks. You're currently muted, but we will unmute you if you have to ask a question. Towards the end of the talk, you can type your questions into the chat box, and then you'll be called on, and you can unmute yourself at that point and ask the question. So with that, I'm going to hand it off to Arun, who's going to introduce our first speaker, Kristen.
Okay. All right. Thank you, Mike. I'm pleased to introduce today's first speaker, Kristen Johnson. Kristen got her PhD with Rob Stalin, where she received excellent training in lipid biochemistry and cell biology. And so with that training, she was a perfect fit for the work my lab does on cholesterol homeostasis here at UT Southwestern Medical Center in Dallas. So I was very glad that she chose to join me here for a postdoc, and she's been here about 2.5 years now. And during that time, she has become quite an expert in cholesterol biology. And today, she will talk about one of her projects where we collaborated with Neil Alto in our Department of Microbiology to find an interesting connection between membrane cholesterol and bacterial infection. So this work was just published recently, and Kristen will present the main findings from this paper. So yeah, take it away, Kristen.
All right. Thank you for the introduction, Arun. I'm very happy to be here today to share this work with you. And I know that these lipid seminars have been a highlight of my quarantine, so I'm really excited to be here today. So before we get started, I want to give a brief introduction to cholesterol homeostasis in cells. The distribution level of cholesterol are precisely regulated in our cells. Most of the cholesterol is in the plasma membrane, and only a small fraction of the cholesterol is in the ER. But the ER contains the cholesterol regulatory machinery. When ER cholesterol levels are low, SCAP traffics SREBP from the ER to the Golgi, where it's processed to release a transcription factor that will upregulate genes involved in cholesterol synthesis, including HMG coreductase, and LDL uptake, including the LDL receptor. These processes will increase the level of cholesterol in the cell, and when satisfied, SCAP will bind to cholesterol and be retained in the ER through retention protein INSIG to prevent further synthesis and LDL uptake. Excess cholesterol in the cell can be moved to a storage form through esterification by ER resident enzyme ACAT. So in the Radhakrishnan lab, we're really interested in understanding how the ER is able to communicate the level of cholesterol with the PM to achieve the ideal level. One solution to this problem is that cholesterol is organized within the plasma membrane. Most of the plasma membrane cholesterol is sequestered through interactions either with sphingomyelin or with other lipids or membrane proteins within the plasma membrane, but some of the plasma membrane cholesterol is accessible to transport to the ER and signal sufficiency. When cells are depleted of sterols, this pool of accessible cholesterol is also depleted and transport is blocked. However, it can be repleted in sterile depleted cells by destroying sphingomyelin with sphingomyelinase treatment, and this will restore signaling to the ER. Currently, we have two toxin sensors to recognize sphingomyelin sequestered cholesterol and accessible cholesterol. They are ALI-A and ALOD4. And these sensors respond to changes in the levels of cholesterol in the plasma membrane. When I deplete cholesterol from the membrane through treatment with cyclodextrin, which extracts cholesterol from the membrane, you can see that accessible cholesterol is rapidly depleted. Harsher treatments can also deplete sphingomyelin sequestered cholesterol from the membrane. When I treat with sphingomyelinase, I can see an increase in the accessible cholesterol level in the plasma membrane and can see that all of the sphingomyelin sequestered cholesterol has been depleted. So today I'm going to share with you a recent collaborative project with the Alto lab in the microbiology department, also at UT Southwestern, and how we used these toxin sensors to study how a interferon gamma stimulated metabolite is able to block Listeria spread. So this work was driven by Michael Abrams, a really excellent graduate student in the Alto lab who recently defended. And he was really interested in finding out whether or not there is a immune response that can inhibit Listeria, which is a gram-positive bacteria that can cause foodborne illness. And we know that it can enter our cells through interactions with our host receptors, where it will then be in a phagosome. To escape the phagosome, it has 3 proteins. One is a cholesterol-dependent cytolysin, and it also has 2 phospholipases that will degrade the phagosome so that the bacteria can replicate in the cytosol of the cell. It can then interact with actin to become mobile and zoom around the cell and eventually penetrate a neighboring cell to continue the bacterial life cycle. So Michael started out with a simple experiment where he added interferon gamma to macrophages, transferred the media from those cells onto intestinal epithelial cells, and then infected the cells with a GFP Listeria strain. He then used flow cytometry to determine the fraction of cells that were infected with Listeria. He found that when he transferred media from untreated macrophages to epithelial cells, about 30% of the cells were infected with the GFP Listeria. After he transferred media from the interferon gamma treated macrophages, the rate of infection dropped to about 4%. So I'm going to skip over a lot of work that Michael did and summarize the findings of his screen. So he knew that the interferon gamma must be upregulating a gene that is secreting a metabolite that's able to be transferred during the media transfer to the epithelial cells to protect against the Listeria infection. And he identified that the gene that was the most potent at inhibiting Listeria infection was cholesterol 25-hydroxylase, which produces an enzyme that converts cholesterol to 25-hydroxycholesterol. And we know that 25-hydroxycholesterol is a potent signaling molecule, not only in metabolism of cholesterol and in immunity. We now know it's broadly antiviral, he has a very potent signaling molecule generated through the cholesterol 25-hydroxylase. So Michael next wanted to investigate which step in the Listeria life cycle 25-HC was inhibiting. He found that the entry was normal, the escape from the phagosome was normal, and also the interaction with actin and the actin-based motility was normal. However, the cell-to-cell spread step was inhibited. So now we have a gene that produces a molecule that's involved in many signaling processes, one of which is cholesterol homeostasis. And we also have the site of inhibition at the plasma membrane, which allowed them to ask, does 25-HC affect plasma membrane cholesterol? And this is where we got involved in the project. I utilized our soluble toxin sensors, OLIA and ALOD4, to detect changes in accessible cholesterol and sphingomyelin sequestered cholesterol after treatment with 25-HC or a different oxysterol 7-alpha-HC. And I observed that after 1 hour, almost all of the accessible cholesterol was depleted from the plasma membrane, while after 8 hours, there was only a very small effect, if any, on the sphingomyelin sequestered pool. 7-alpha-HC had no impact on either the accessible or sphingomyelin sequestered cholesterol pools. We had this very robust finding, but we weren't sure how it correlates with the inhibition of Listeria. spread in cells. So Michael and I repeated the same experiment with our different assays to determine both accessible cholesterol level and the percent infection inhibition. So we set up cells in either a cholesterol poor condition or a cholesterol rich condition, and then treated cells with various concentrations of 25HC for 16 hours. After that, I added LRD4 to determine the percent bound to the surface of the cell, and could see that in the cholesterol poor condition, accessible cholesterol was rapidly depleted by 5HC. Similarly, after accessible cholesterol was depleted, the percent infection inhibition of Listeria was increased. Similarly, in the high cholesterol condition, we can see that while more 25HC is required to effectively deplete the accessible cholesterol from the membrane, we see the same trend that when accessible cholesterol is depleted, we achieve inhibition of Listeria spread. So if accessible cholesterol is truly important for Listeria to achieve cell-to-cell spread, then replenishing it should reverse this effect. So we used two different techniques to replete the accessible cholesterol from the membrane. The first was with treatment with sphingomyelinase. So under normal conditions, we can see that both accessible cholesterol and sphingomyelin sequestered cholesterol are present on the plasma membrane. After treatment with sphingomyelinase, we see an increase in accessible cholesterol and at the expense of the sphingomyelin sequestered cholesterol pool. After treatment with 25HC, all of the accessible cholesterol is depleted, while sphingomyelin cholesterol is unaffected. Then a sphingomyelinase treatment can restore the accessible cholesterol level, again, at the expense of the sphingomyelin sequestered cholesterol pool. Importantly, the sphingomyelinase treatment after 25HC also reverses the protective effect that 25HC had against Listeria spread. The second approach that we used to replete cholesterol was with exogenous addition of cholesterol complexed in cyclodextrin. So you can see that under normal conditions, accessible cholesterol is present, which is depleted by 25HC. Addition of cholesterol was able to restore accessible cholesterol levels. And importantly, we see that, again, addition of exogenous cholesterol was able to reverse the protection provided by 25HC, suggesting that accessible cholesterol is truly important for Listeria cell-to-cell spread. So next we asked what pathway is 25HC acting through to deplete accessible cholesterol from the plasma membrane. We know that when ER cholesterol levels are low, we have stabilization of HMG coreductase, we have low ACAT activity, and we have SREBP transport and processing to upregulate genes to increase the level of cholesterol in the cell. When ER cholesterol is high, we get degradation of HMG coreductase, SREBP is retained in the ER, and we have high ACAT activity to move excess cholesterol into the storage form in lipid droplets. And we also know that regardless of high or low ER cholesterol levels, oxysterols can override this normal process, where regardless of cholesterol level, SREBP is retained in the ER, and regardless of cholesterol level, ACAT activity is high and will move cholesterol into lipid droplets for storage. So to see whether one pathway was responsible for the effect that we were observing at the plasma membrane, I expanded the oxysterols that I was working with. So I included a A-ring oxysterol for beta-HC, a B-ring oxysterol 7-alpha-HC, and then 3 additional isooctyl side chain oxysterols, 20-alpha-HC, 22-R-HC, and 27-HC. First, I looked at the ability of each of these oxysterols to deplete accessible cholesterol from the plasma membrane using the ALOD4 assay. After treatment with 4-HC, we can see that there is no change in the level of full cholesterol at the plasma membrane. Similarly, 7-HC does not change the level of accessible cholesterol. However, we saw that 20-HC was able to deplete accessible cholesterol, similarly to 25-HC. Surprisingly, a different isooctyl side chain, oxysterol, was not able to deplete accessible cholesterol. And similar to 25-HC, 27-HC also depleted accessible cholesterol. Next, I looked at the ability of each of these oxysterols to control SRE-BP behavior. So in order to observe any changes in this, I first depleted the cells with cyclodextrin to force SRE-BP into the nuclear form, and then I added back increasing amounts of oxysterol to each. After treatment with 4-HC, SRE-BP was not recovered from the nuclear form to the precursor form. After treatment with 7-HC, we can see recovery of SRE-BP. Similarly, 20-HC, 22, 25, and 27 were all able to recover SRE-BP. So the important finding from this set of experiments is that the effect of cholesterol depletion is dissociable from SRE-BP2 control by the oxysterols. So even though 22R is able to control SRE-BP, it's not able to deplete accessible cholesterol from the plasma membrane, which made us think that this phenotype that we were observing of rapid cholesterol depletion was acting through a different pathway. So we next turn to ACAT as a potential pathway for the depletion of the accessible cholesterol by these oxysterols. And I utilized a specific ACAT inhibitor, Sandoz 58035, for this assay. So under normal conditions, we can see accessible cholesterol is present, but after treatment with either 20-HC, 25-HC, or 27-HC, accessible cholesterol is depleted. However, the oxysterol treatment in the presence of the ACAT inhibitor slowed the depletion of the accessible cholesterol. We can see there's still a very similar amount compared to no treatment after inhibition of ACAT. So unlike the pattern that we observed with the SRE-BP, we can see that there is a perfect match between depletion of accessible cholesterol and a dependence on ACAT activity for this depletion. Importantly, this ACAT inhibition also was able to reverse the protection that 25-HC provides against infection. So you can see that when cells are treated with the inhibitor alone, there's no effect on Listeria spread. 25-HC significantly decreases the percent of infected cells, while treatment with the ACAT inhibitor prevents infection. But as we continue to do these experiments, we observed that after short time periods, the ACAT inhibitor was a perfect inhibitor of accessible cholesterol that blocked accessible cholesterol depletion by 25-HC compared to the 25-HC alone. But after longer time points, we could see that some accessible cholesterol was being depleted even though ACAT was inhibited, which made us think that this mechanism of action includes more than one pathway. So we went back and investigated whether or not SRE-BP does have a role in the lasting immune effect against Listeria. And we utilized a cell line that constitutively expresses the nuclear SRE-BP2 and is resistant to control by oxysterols. So using the LRD4 assay, we can see that 25-HC is unable to completely deplete the accessible cholesterol from the plasma membrane. And if we compare this to the wild type cell line CHO7, you can see that while CHO7 accessible cholesterol is completely depleted by 4 hours, we still have about 40% of the accessible cholesterol left in this mutant cell line. And this translates into the infection model. We see that in the mutant cell line, 25HC is no longer able to block Listeria spread, which indicated to us that the SREBP, while it's not responsible for the rapid depletion of accessible cholesterol, it's still important to sustain the response and keep plasma membrane accessible cholesterol low. So that brings us to our summary model, where we found that interferon gamma activated macrophages upregulate cholesterol 25 hydroxylase, and this produces 25 hydroxycholesterol, which can circulate and enter epithelial cells to activate ACAT. And when ACAT is activated, the accessible cholesterol in the plasma membrane is mobilized from the plasma membrane to the ER, where it is then esterified by ACAT for storage in lipid droplets. So we believe that this is the rapid, fast response driven through 25HC, but there's also an important prolonged response to maintain low accessible cholesterol, and that's through interaction with the SREBP SCAP and SIG complex to retain SREBP in the ER and prevent the replenishing of accessible cholesterol while the infection is ongoing. So we still have several outstanding questions, including we still don't understand what the mechanism of cholesterol movement from the plasma membrane to the ER is. Also, we do not know why Listeria requires accessible cholesterol for cell-to-cell spread. And we are curious to understand whether accessible cholesterol regulation is a broadly antibacterial or antiviral defense in the immune scheme. So with that, I'd like to thank my mentor, Arun, as well as an excellent technician that I've been working with in Arun's lab, Danja. I'd also like to thank our amazing collaborators, Neil and Michael. It's been a real pleasure working on this project with you guys. And I'll certainly miss Michael now that he's recently defended. But fortunately, Neil has several other excellent trainees in his lab that we can continue to work with. Thank you to Mike and John for giving me the opportunity to share my work today. And thank you to the funding. So I would be happy to take any questions if you have them.
Okay, thank you. Thank you, Kristen. We have some questions. So first question is from Clarissa Maya Montero. If you could please unmute and ask your question.
Yes. Thank you, Kristen. It was a wonderful presentation, an enormous amount of work. But I was missing my dear lipid droplets. Did you measure? Did you stain for that?
Yes, we did some staining of the lipid droplets. It's in one of the supplemental figures. Unfortunately, this is a very nice assay. So 25HC on its own will not induce giant lipid droplets, but it does increase the lipid droplet number, and there's lots of small lipid droplets observed compared to the vehicle treated samples.
Yeah, okay, thank you.
Okay, next question, Mike.
Beautiful talk, Kristen. My question is sort of, if I remember right, Listeria has a sphingomyelinase, a bacterial one, and you also see it in Bacillus cereus and Staphylococcus. So do you think that what's the role of the Listeria sphingomyelinase? I guess, did you guys look into that? Because you're treating with sphingomyelinase, I guess, exogenously.
Yeah, we did not look into the role of the bacterial sphingomyelinase. My hypothesis would be that it would be important for the escape from the phagosome, especially when after the first cell cycle, when there's a double membrane, perhaps it needs to degrade other lipids to generate accessible cholesterol for the LLO CDC for escape.
I see. So there's no like implication that the listeria would use it for that the last step to actually penetrate into other cells to overcome this.
Not that I'm aware of, but that would be really interesting to look into.
Okay. Since there's no other questions, I'll ask another one actually. I'm just curious. I mean, I know Yusuf Anoon's group uses sphingomyelinase to look at ceramide signaling. How do you you have beautiful effects on cholesterol accessibility. How do you discriminate that ceramide is not playing any role? Have you thought about using a ceramidase to quickly get rid of the ceramide that's generated by the sphingomyelinase?
We have not tried that treatment before, and I will say that the treatment with the sphingomyelinase is very short because we are trying to avoid having off effects because of the ceramide generated, but we have not used the additional reagents to degrade the ceramide. Be interesting to see if there's any change in what we see.
Thank you.
Okay. We have another question from Denisa, and then from
Yeah.
Denisa.
Yeah, hello, very nice talk. I wanted to ask, did you guys have a look at oxysterol binding proteins and how they may affect,
No, that's a great question. We have not looked at that yet, but I hypothesize they may have a role in the cholesterol transport. So that's something that we still don't understand whether or the mechanism of mobilizing the accessible cholesterol from the plasma membrane to the ER after the oxysterol treatment. So that's definitely a future direction.
Okay, so since this is a cholesterol talk, it's not surprising that Mike has a question. Go ahead, Mike.
Yeah. My question is, so sphingomyelinase, mobilizes cholesterol initially. It must initially increase the concentration in the plasma membrane before the cholesterol then gets transported to the ER, so where it's esterified. So the question is, I guess your assay, you can't really time it well enough to see whether there's a transient time where you actually increase the spread before you decrease it. Or if you use an ACAT inhibitor plus sphingomyelinase, does that increase the resistance to 25 hydroxycholesterol? In other words, does it change? Imagine using sphingomyelinase, and if you can't esterify the cholesterol, what happens to it?
That's a great question. I actually haven't done that experiment yet, but I hypothesize that the level of accessible cholesterol that we would measure in the plasma membrane after the sphingomyelinase treatment would be much greater than what we see. So normally we see a marginal increase, but we do know from previous work that after sphingomyelinase treatment, that the cholesterol that is liberated from sphingomyelin is able to transport rapidly to the ER for esterification. So what we're measuring is probably a minor amount of the total that is liberated. So if ACAT is inhibited, and then I would think that would dramatically increase the accessible cholesterol in the plasma membrane and maybe promote the listeria cell-to-cell spread. So it would reverse the protection.
If ACAT is inhibited, I mean, theoretically, the cholesterol would still go to the ER, but now it must go back to the plasma membrane again. That would be interesting to study sometime, independently of listeria.
I agree.
Oh, let me just say one more thing. Just a word of congratulations to you and Neil and Michael. I think this is really not to mention a room, but this is a really beautiful series of studies. I mean, really really impressive. So congratulations.
Thank you, Mike.
Okay, well, can't really beat that ending, so I guess we'll end here. Thank you.
Fantastic. So Barbara, you want to can hand it over to Barbara and she can introduce.
Okay, there we go. All right, thank you for that. It is my really sincere pleasure to introduce Dr. Dequina Nicholas to the group today. Dequina did her PhD at Loma Linda University in Southern California in the Division of Biochemistry before she joined my lab then at Boston University, where she did 2 years of postdoctoral work under my guidance. And the work that she'll be presenting today was based on that tenure at Boston University. And I want to emphasize the fact that this project really was Dequina's brainchild. I just had the great pleasure of going along for the ride. She also hung in there through my move to the University of Kentucky, where we continued to work on getting this paper out into the public domain. And while she was doing that, she completed or is continuing to complete a second postdoctoral fellowship with Dr. Mark Lawson and Pam Mellon at the University of California, San Diego. And I have to finish by congratulating Dequina. She will be starting her independent lab at the University of California at Irvine, starting the mid to late 2021. So congratulations to her on that. So Dequina, take it away.
Okay, awesome, great. Thank you so much for the introduction. And I also want to thank the organizers, Mike and John, for giving me the opportunity to present and share this work. So type 2 diabetes is characterized by chronic high blood glucose. And the prevalence of this disease is increasing at an alarming rate worldwide. It's projected that by 2030, just in 10 years, that 400 million people are going to be impacted by this disease. In light of the current pandemic, it's really important to understand the etiology of type 2 diabetes, as those with type 2 diabetes have a higher risk for severe disease once they contract COVID-19. So we know that in type 2 diabetes, the underlying etiology is multifactorial. There are many risk factors. For example, lack of exercise or sedentary lifestyle, genetics such as family history, race, your ethnicity. Also, increasing age is a risk factor associated with type 2 diabetes. But by far, the most predictive factor for the onset of type 2 diabetes is overnutrition leading to obesity, defined by a BMI of greater than 30. In particular, we know that overnutrition leads to chronic inflammation. That is a process that has been shown multiple times in various publications to be important in the onset and development of insulin resistance in mouse models. And in general, chronic low-grade tissue inflammation, specifically in the visceral adipose tissue, is linked to obesity and development of type 2 diabetes in both mouse and human. However, when clinicians or studies have targeted inflammation in type 2 diabetes, a lot of these interventions have fallen short. And why might these reasons be? One reason could be that the focus of the field is largely on the cytokines that were first discovered to be associated with type 2 diabetes. Namely, this would be tumor necrosis factor alpha, interleukin 6, and interleukin 1 beta. These three cytokines are secreted at very high levels and easy to detect. However, just because they were the first to be discovered may not necessarily mean they're important for type 2 diabetes inflammation. The other issue may be that the current gold standard mouse model of obesity, or the high-fat diet mouse, is known to have an inflammatory process that is different from what we see in humans with type 2 diabetes. In mice that you feed a high-fat diet, you see that the inflammation is dominated by what we call a Th1 T cell type inflammation. However, in humans, there have been several labs that have shown that Th17 inflammation is what is occurring. For example, Sam Klein, Karen Clement, and of course the Nikolaichik laboratory have all published that Th17 cells are a defining characteristic of human type 2 diabetes. In a 2011 publication from Dr. Nikolaichik's laboratory, we see that there are increased amounts of Th17 T cells circulating in subjects with type 2 diabetes as compared to subjects who do not have diabetes. We've also shown that not only are there increased amounts of these Th17 pro-inflammatory cells, but that these cytokines, if you treat these cells, that if you treat them in vitro, also secrete increased levels of Th17 cytokines. So these would be interleukin 17A and F and IL-21. And together we say that all these cytokines secreted by Th17 cells produce what we call a Th17 cytokine profile that is very characteristic of human type 2 diabetes as opposed to mouse type 2 diabetes. So, but what we don't know is why do we have a Th17 cytokine skewing in human type 2 diabetes? What we do know from a growing body of literature is that immune cell function, such as cytokine secretion, is supported by different metabolic programs. So just to be very simplistic, I have for you an example such as effector T cells rely heavily on glycolysis in order to support their programs in cytokine secretion. On the other hand, regulatory T cells are supported by fatty acid oxidation as opposed to glycolysis. And then we kind of see this theme with a lot of different subsets within the immune system. So it follows that if these immune cells have certain metabolic programs by which they use to support their function, then if there's a change in the nutrient environment or the metabolic milieu that is around these cells, the fuels that are available, that it may impact the metabolic program and therefore impact their function. So that brings us to this overall question. In type 2 diabetes, does metabolism drive Th17 inflammation that we know is associated with human type 2 diabetes? So there are three major circulating fuel sources that we know in humans correlate with insulin resistance and are elevated in people with type 2 diabetes. The first is going to be glucose, which is very obvious. The second are elevated free fatty acids, particularly saturated fatty acids. And the third are branched chain amino acids. We believe that in type 2 diabetes, this overnutrition and these excess fuel sources may impact cellular metabolism and therefore impact the function of immune cells such as proliferation, cytokine secretion, or differentiation. And then for this paper, we really focused on cytokine secretion and what is impacting the Th17 cytokine profile. So we hypothesized that these fuel sources, or the change in the metabolic milieu that we see with overnutrition, and as we mentioned, impact T cells or peripheral blood mononuclear cells within subjects with type 2 diabetes, impact their metabolism, and therefore support pro-inflammatory cytokine secretion. So through a series of experiments that I don't have time to share with you today, we're actually able to rule out branched chain amino acids as impacting metabolism in immune cells in persons with type 2 diabetes. So of course, the next obvious case was to look for glucose or fatty acids. And in order to analyze the metabolism in response to these two major pathways, we use a technique called extracellular flux analysis, which can measure two major energy pathways of the cell. As you know, fatty acids, once they come into the cell, go through a series of biochemical reactions that cause their metabolism. And then we see that with fatty acid transport, which we'll go into a little bit in more detail further in the talk, we have fatty acid oxidation, and some of these metabolites go through the TCA cycle in order to generate ATP. The TCA cycle, electron transport chain, require the use of oxygen, which we can measure the oxygen tension, and therefore get the oxygen consumption rate, which would be a proxy for looking at aerobic respiration within these cells. Simultaneously, we can also measure the acidification rate or extracellular acidification rate, ECAR, of these cells based on the amount of lactate that is being produced from the cells. And therefore, we can get a measure of glycolysis and a measure of aerobic respiration in immune cells from subjects with type 2 diabetes. So what we did was take peripheral blood mononuclear cells, or PBMCs, from here on out, from subjects with and without type 2 diabetes that were age and BMI matched. This was really important. Then in vitro, we stimulated these cells with T cell targeted stimuli using antibodies against CD3 and CD28, and then performed the mitochondrial stress test. You don't really need to know anything about a mitochondrial stress test to see that this OCR is not different at all between the non-diabetic PBMCs and the type 2 diabetic PBMCs. However, when we look at the extracellular acidification rate, our proxy for glycolysis, it's pretty obvious that we have increased glycolysis in the cells from subjects with type 2 diabetes. This lets us know that the immune cells from subjects with type 2 diabetes are indeed more glycolytic than those from non-diabetics. we were actually able to verify these results using a lactic acid assay. And so we know that this extra acidification in the media was caused by lactate. So this data supports the idea that glucose may be fueling metabolism within cells from subjects with type 2 diabetes. And we really wanted to know, therefore, if glucose was driving the cytokine production or Th17 cytokine production we see in immune cells from persons with type 2 diabetes. So to do this, we once again went back to our model of taking PBMCs, stimulated them in vitro, but this time we did it in the absence of glucose. These cells were glucose starved. We then used the conditioned media and measured 25 different cytokines by multiplex. After measuring these cytokines, we used a supervised clustering method called partial least squares discriminant analysis, or PLSDA, in order to ask the question, what happens to cytokine production if we remove glucose from the environment? So what this type of analysis does with our data is produce a representation matrix of data that's visualized by a single point. So for example, this point here represents the 25 different cytokines that are produced by a single patient. Then what the PLSDA algorithm does is find the multidimensional orientation that will maximize separation between groups on a single axis. And in this way, we have what we call a latent variable. And then we're able to see what variables are helping to contribute to differences that we see between, in our case, non-diabetic cytokine production and type 2 diabetic cytokine production. So for this experiment, what we would expect is that if glucose is driving type 2 diabetes inflammation, then if we remove glucose, then we would no longer be able to tell the difference between non-diabetics and type 2 diabetics. So we expect some overlap. However, when we actually analyzed our data, we found the opposite, which was really surprising. We were able to get a really clear distinction between the cytokine profiles between subjects with and without type 2 diabetes. And this data was able to tell us two really important things. One, that PBMCs from these two cohorts have different abilities to utilize non-glucose fuels. And two, that this inflammation is not driven by glucose. If it's not driven by glucose, then what is it driven by? So of course, of these three options I presented you for fuels, the last one is free fatty acids. And then in order to determine if lipid metabolism is playing a role at all, we went with a really broad approach, and we performed an RNA microarray for PBMCs from non-diabetics and type 2 diabetics. And we confirmed that there's genes involved in lipid metabolism, particularly fatty acid or lipid transport into the mitochondria that were dysregulated in type 2 diabetes. So briefly, I'm just going to go over mitochondrial fatty acid transport because it's important for our experiments going forward. Fatty acyl-CoA derived from your free fatty acids, in order to be metabolized within the mitochondrial matrix, first has to undergo conversion by carnitine palmitoyltransferase 1, or CPT1, into fatty acylcarnitine. This is the rate limiting step for mitochondrial fatty acid transport. This fatty acylcarnitine then must be actively transported by carnitine acylcarnitine translocase, or CACT, in order to get into the inner mitochondrial matrix to undergo beta-oxidation. So we decided to look at these two major proteins important for mitochondrial transport, CPT1 and CACT, in our PBMCs from subjects with and without type 2 diabetes. When we looked at CPT1A, we saw that CPT1A was higher in PBMCs from subjects with type 2 diabetes. Conversely, with CACT, we found a reduction in the protein expression by Western blot. When we represented this data as a ratio of CACT to CPT1A, we found that this ratio was reduced in PBMCs from subjects with type 2 diabetes. And what this ratio tells us is that the lower the ratio, the less capacity these cells have to transfer the fatty acylcarnitine that is made by CPT1A into the inner mitochondrial matrix for beta oxidation, meaning that they most likely have inefficient beta oxidation. So just to recap, in cells from people with type 2 diabetes, have increased CPT1, decreased CACT, which likely means you have increased fatty acylcarnitines that are not being oxidized within the cell. So we believe that CPT1 activity was important for the cytokine profile we saw in type 2 diabetes. But to test this, we wanted to use a small molecule inhibitor atomoxer, which would block the activity of CPT1 and therefore mimic what we see in this model. So what we did was go back to that original experiment where we stimulated our non-diabetic and type 2 diabetic PBMCs with T-cell directed stimuli for 40 hours. We then measured the conditioned media for multiple cytokines by multiplex and performed PLS trials. So unlike what we saw with the glucose experiment, where we got rid of glucose, but we could still differentiate the two groups, when we added atomoxer to block CPT1A activity, we were no longer able to distinguish between the two groups. So what this told us is that CPT1A activity was indeed important for type 2 diabetes associated Th17 cytokine profile. So to confirm this, what we did was rank our cytokines by what we call the variable importance and projection score, or VIP score, which is telling us which cytokine is most important for differentiating between atomoxer treated or not treated, regardless of their diabetes status. So what we're able to see is that if they have a VIP score greater than 1, then they are important for distinguishing atomoxer treated from control. And what we found is that Th17 cytokines, highlighted in red, and then cytokines which support Th17 cells, highlighted by red and black stripes, are important to distinguish atomoxer treated from our control cells. So therefore, we're able to conclude that CPT1A activity is important for Th17 cytokine production. So in a really simple way of putting it, treating with atomoxer will reduce the amount of Th cytokines secreted from peripheral blood mononuclear cells. So we illustrated, once again to recap, that CPT1A is high in T2D PBMCs, and that CACT is low. In this scenario, type 2 diabetic cells would have a reduced capacity to transport fatty acylcarnitine into the inner mitochondrial matrix for beta-oxidation. And therefore, we speculated that the fatty acid metabolite, fatty acylcarnitine, is accumulating in the extracellular space and in the cytoplasm and may initiate lipid signals that could downstream have effects on Th17 cytokines. So what we wanted to do was test whether or not beta oxidation is important for Th17 cytokine secretion. And to do this, we wanted to use a beta oxidation inhibitor, trimetazidine. So we did this in vitro. This time, instead of using PBMCs from non-diabetic or from type 2 diabetic donors, we stimulated PBMCs from lean, healthy donors. So these are healthy PBMCs. We stimulated them either in the presence or absence of trimetazidine to block beta oxidation. What we found is that when we block beta oxidation, we actually get an increase in secretion of Th17 cytokines, IL-17F and IL-17A, but not of cytokines associated with Th1 T cells, such as IL-6, necrosis factor beta, or with Th2 cells. So these data agree with the hypothesis that acylcarnitine support production of Th17 cytokines. We have not directly tested this. To directly test whether the acylcarnitines could indeed induce Th17 cytokine secretion, we decided to add exogenous fatty acylcarnitine to PBMCs from lean, healthy donors. In addition to adding this exogenous fatty acylcarnitine, we also knocked down the carnitine acylcarnitine translocase, or CACT. This was important because we did not want this exogenous fatty acylcarnitine we're adding just to simply be transported into the mitochondrial matrix and then get oxidized. Instead, what we wanted was for this fatty acylcarnitine to accumulate the high levels as we presume they do in cells from type 2 diabetic donors. So for this experiment, after we knocked down CACT and added exogenous acylcarnitines, we performed intracellular cytokine staining for IL-17A and IL-17F and analyzed them by flow cytometry. On the left are representative dot plots, and then we see that if we only knock down CACT, we don't see a change in IL-17F production. If we only add the acylcarnitine, palmitoyl L-carnitine or C16 L-carnitine, that we also see no change. However, when we combine the two by adding exogenous acylcarnitine and knocking out CACT, we see an increase in IL-17 positive producing cells. And this is quantified here on the right. What's interesting to note is that if we do the same experiment with short chain or medium chain fatty acids, which don't need this transport machinery in order to go into the mitochondrial matrix, then you actually have no effect. So this only works with long chain fatty acids. And if we do the converse and take type 2 diabetic PBMCs, knock down CPT1A, we get a reduction in IL-17F positive cells. So these data show us that the Th17 cytokine production, or Th17 positive cells, are impacted by high levels of acylcarnitine. So altogether, what I've been able to show you today is that compared to non-diabetic PBMCs or cells, there is disrupted lipid flux in immune cells from subjects with type 2 diabetes. You have increased levels of free fatty acids, which has been shown in several studies. We think that this disrupted lipid flux is mainly contributed or due to a high level of CPT1, low level of CACT, which therefore prevents acylcarnitines from being effectively transported into the inner mitochondrial membrane space. So therefore you get reduced fatty acid oxidation. This excess long chain fatty acylcarnitine is then, through a mechanism that is under investigation now, somehow increasing the production of Th17 cytokines. So this is very important because we know that in the clinic, better glycemic control is generally the goal of type 2 diabetes interventions. And what we have done is demonstrate that glucose is not the dominant activator of Th17 mediated inflammation. So this raised the concern that once we manage blood glucose in type 2 diabetes, why are there still other comorbidities or problems that happen down the line? And this could be because we need to also manage the lipids or the changes in lipid flux that we see in subjects with type 2 diabetes. So with that, I'd like to thank my mentor, Dr. Barbara Nikolajczyk, and the whole entire team in Barb's lab who helped all this work come together, and also our collaborators, and then lastly, our funding. And once again, I'd like to thank the organizers of this seminar series for inviting me to give a talk, and I will take questions at this time.
Great, thank you so much, Dequina. Your first question is from Raphael. Go ahead, Raphael.
Hi, Dequina. There was can you hear me?
Yes.
Okay, that was a beautiful talk. I have a twofold question, if you will. So the first one is, it's more of an observation. Usually when we think of a rate limiting step for long chain fatty acid import into the mitochondria, we think CPT1, right? So It's kind of amazing that you're actually seeing an effect on the CACT transporter. Do you have any ideas on like how that is actually being regulated in the type 2 diabetes setting? And another question that I have is, do you think it's any fatty acyl conjugated to carnitine, like just a total accumulation of that is causing the phenotype? Or do you think it's specific fatty acyls like a C16? You tested C16, but do you think any other specific ones would be more involved?
Okay, so starting off with your first question, yes, we don't normally see CACT as being regulated, especially for mitochondrial transport. And at least from our data, it seems like majority of that change that we're seeing is actually being driven by the levels of CPT1A, even though we do see less CACT. If I were going to comment on what I think might be a mechanism that's regulating it, is that the body's getting mixed signals in type 2 diabetes. Normally you have, say, your fasting state free fatty acid. In type 2 diabetes, you're seeing high levels of both, which can be confusing to the cell. So that might be the beginning of a mechanism whereby you're regulating the expression of CACT. But like I said, we have not yet tested this. For your second question, I did do some preliminary experiments with longer chain fatty acyls, and it seems like there's basically a sweet spot, which is the size chain fatty acids that you'd normally see within the diet that's being metabolized normally within the body, like your C16, C18s. We didn't check to see if there's a difference, whether or not, say, it's saturated or polyunsaturated, and if that's going to have an impact. But we do know that it needs to be long chain fatty acids as opposed to short chain and medium chain.
Awesome, thank you.
Mike, you have a question?
Yeah, Dequina, fantastic talk. So I'm as a structural biologist, can you explain the significance of the PBMCs? And if I can ask the second question, so based on your data, what new kind of therapy do you imagine that might be useful in type 2 diabetics? Those are my two questions.
Okay, great. Those are great questions. So the importance of PBMCs is that the inflammatory profile that we see from PBMCs can really well reflect the type of inflammation that we see going on in adipose tissue. And this has been shown by several laboratories. It's been already established that the inflammation we see in adipose tissue is important for development of insulin resistance. So this gives us a really good proxy that's easily accessible from patients, where we can have repeatable experiments. And on top of that, the peripheral blood mononuclear cells is what we call a mixed or dirty model, which is all these different immune cells interacting together, which we have found that these interactions between the immune cells are important for the phenotypes that we see. So it's like the opposite of doing a reductionist model. So that's why we like to use the PBMCs. And then specifically for this model, we need this mixture in order to get the metabolic phenotype that we see from stimulating the T cells. So that's why PBMCs are important. The second thing for within the clinic, I think one of the things that would be important to start doing initially is maybe not so much a new drug or new intervention, but to start monitoring the lipids as importantly as we monitor the glucose with management regulation. There's already plenty of drugs for lipid management that are used in the clinic. So I think focusing on both glucose and lipid management is going to be important moving forward instead of just opposed to glucose management.
Exciting. Thank you.
Alan Addy has a question. Go ahead, Alan.
Yes, very nice talk, very nice science. Is the effect on IL-17 an effect on its production or its secretion?
So we didn't directly measure whether it's the production or secretion. We know for a fact that there's more or less being secreted because of the inhibition of, say, the beta-oxidation, because our output has always been secretion. But to parse apart whether or not we are impacting the actual like transcription and translation of the cytokines, we have not yet measured. And then from my perspective, I would hypothesize that we are impacting the actual production of the cytokines. For the most part, how much is getting secreted is directly related to how much is being made within the cell, but it is possible that we are impacting the ability of the cells to secrete it. And then, but I think that if it really were a secretion issue or a problem, we would have seen that turn up in our flow cytometry data where we stained for the intracellular cytokines. We would have seen a difference between what we see with our secreted data and with our internal data.
Well, yeah, just to follow up. So there's quite a lot of work in pancreatic beta cells on fatty acid effects on insulin secretion. And you wouldn't see a change in content because it's only a small fraction that gets secreted. But there's a beautiful body of work by Mark Prentke that you might want to look at, where he's taken it all the way to identifying monoglycerides as the key intermediate in mediating the effect, the interactions with a snare protein called MUNC13. And there may be a similar mechanism in the T cells.
Thank you. I mean, that's actually a really great clue because we're trying to figure out where to go from here with the mechanism and how these lipids are actually impacting the cytokine production and secretion.
Yeah, thanks for that suggestion, Alan. I'd like for Pablo now. Pablo has a question.
Yeah. Hello, this is Pablo, Mount Sinai School of Medicine. Fantastic talk, Dequina. I have a question. Have you considered to modulate carnitine levels, maybe by targeting OCTN2 transporter to see if you have an effect when you modulate carnitine levels? Thank you.
Can you repeat the second half of that question, please?
Yeah, but by targeting OCTN2, the carnitine transporter.
Oh, OCTN2. No, actually, I haven't considered it, but there was another postdoc in the lab that was actually looking into that transporter. We did some lipidomics and then pathways involving that actually did come up. So I think there might be there's a probability that it could play a role, and I think that'd actually be a really good idea to do.
Suzanne Barbar has a question.
Thanks. That was a really nice talk, Dequina. You mentioned there's a genetic component to type 2 diabetes. I wonder, are there other Th17 and metabolism-driven diseases that you think would have this same underlying mechanism?
I'm actually, yes. I love this question. So I've actually been a little bit curious on the side about psoriasis and the Th17 dominant inflammation that we see there. And I've seen in several studies that there is like lipid dysregulation that could like cause flare, things like that. So I think there's a probability that this pathway is not limited only to type 2 diabetes. And it'd be really interesting to see any comparison and do like a GWAS between people with type 2 diabetes and Th17 inflammation, and also compare other inflammatory diseases that are Th17 driven, specifically autoimmune diseases. I do think there could be some similarities in the pathways that are being regulated.
And then our final question will come from Paul Watkins. Paul, go ahead. Oh, Paul, you don't have audio. Sorry. So I will read his question. He is wondering what is going on with CPT2 in your system.
So I'm sorry to inform you, but we do not know what is going on with CPT2. We did not measure it. And then also in our microarray, CPT2 was not in the array. So even though I think it's really important to look at and to know, we actually did not measure it. So we don't know what's going on with CPT2.
All right, so with that, we are at time and I am not seeing any additional questions. I can wait just a minute or hand it over to John to wrap up.
Yeah, thank you, Barb. I just want to thank everyone for attending, and let's give another round of virtual applause to our speakers, Kristen and Dequina. You guys did fantastic. I always love learning about new science and new lipids. So thank you guys again. And thanks everyone for attending. We'll see you guys next week.